So I want to talk about the role of glutamate in spreading seizures. So first of all, seizure is excessive and abnormal brain cell activity. On the left-hand side, you'll see um, just an example of EEG where you can see brain activity, but it's not abnormal. It's more like they're just showing normal brain activity. So we also need to understand what glutamate is. So glutamate is a non-essential amino acid, also acts as a neurotransmitter. It's also called a king of neurotransmitter. That's because it's responsible for more than half of the synapses in the brain. So that's why they call it the king of neurotransmitter, because most of the synapses in the brain are from glutamate receptors. It can be only obtained through diet, but it cannot cross the brain barrier, so it must be synthesized in the brain with the help of alpha ketoglutarate, which is an intermediate product in citric acid cycle. Glutamate is also a major hypothalamic excitatory neurotransmitter. When it interacts with the receptors, it causes changes in action potential, which helps in building memory related signals, neural development, and synaptic plasticity. So synaptic plasticity is basically the synaptic changes that happen in response to like an experience, which is an important function so we can learn and memorize. There are two types of neurotransmitter receptors, one being metabotropic. They act through a second messenger. It can be either excitatory or inhibitory action. Another one is ionotropic. Ionotropic, they have three main receptors that activate NMDA, AMPA, and kyanate receptors. It allows positively charged sodium ions to postsynaptic neuron by depolarizing the neuron, making it more likely to fire an action potential. And another thing I want to mention, NMDA receptors have characteristics that make them unique for synaptic plasticity. So this slide, you can see the journey of glutamate to the brain, or glutamate synthesis and glutamate cycle. So basically, glutamate is synthesized in the central nervous system as part of the glutamate cycle by the enzyme glutaminase. Glutamate is removed from synaptic cleft via protein. Furthermore, the protein carries glutamate to neurons and glial cells. And the complete cycle, you can see it here on the left-hand side using the arrows. And this is an ongoing cycle, as you can see from the arrow. So just to recap, glutamate can be synthesized from glucose in the Krebs cycle, and alpha ketoglutarate, which is an astrocyte. Then glutamate is converted to glutamine by enzyme glutamine synthase. Then glutamine is released as a neurotransmitter, and once it is released, it is taken up by a neuron. Through glutamine transporter, the glutamine is taken up by a neuron, and then the neuron converts glutamine to glutamate by glutaminase. And this slide, I'm going to talk about the blood-brain barrier and glutamate. So excitatory amino acid transporter, otherwise known as EATS1, in abdominal membrane moves glutamate from extracellular fluid to endothelial cell where glutamate can diffuse to blood. Next, EAT2, which is a transporter for glutamate, is found on neuron astrocytes and endothelial cells. Transports glutamate, sodium, hydrogen in and out of the cell. You can see it here on the left-hand side. Also, potassium in exchange. So seizure and electroencephalograph. So seizure is detected using electroencephalograph. Electro meaning electrical activity, and then encephalo is the brain, gram is just picture, or sometimes they call it encephalograph. So graph, as you can tell, it's a picture of the electrical activity of the brain. So there are a lot of electrodes that are placed on the head in order to pick up brain activity. And then when that seizure happens, the electrodes pick up those activity from the brain. So it can be quite a complicated process to put those electrodes, especially on newborns or babies, or patients with altered mental status. So, as stated before, seizure is a sudden change in the brain's electrical activity. As a result of this change, it kind of temporarily affects the individual's behavior and actions. This could be before the seizure, ictal state, or post-ictal state. Ictal meaning the seizure happening, or the duration of the seizure. Post-ictal is after the seizure. They could still have their behavior and actions could still be different even after a seizure. So it can be quite interesting to see how some of the patients act following a seizure, because it's not really themselves. Sometimes you'll see them acting a little bit different or odd. That's because they're in a post-ictal state where they could be experiencing psychosis. So there are two types of seizures. One is primary generalized, where the entire brain is affected. And then the other one is partial seizure or focal seizure, where abnormal firing in one region happens and then it just stays there, doesn't generalize to the whole brain. Sometimes seizures could start as focal, but then it could become primary generalized. It could start at like one part of the brain and then spreading to both sides. So, as stated before, EEG is used to test the brain activity, and usually the diagnosis of epilepsy is having more than 2 seizures. There's usually hard to diagnose someone if they're having just one seizure. It's hard to tell why it's happening, but if it keeps coming back, that's when we give this term epilepsy. So yeah. Here on the left hand side, where you see the green kind of background, it's an example of an EEG where the patient is showing a lot of muscle activity. So this could be an example of where the patient is either doing some kind of activity like exercise or moving around, or sometimes even thinking, if they're thinking too hard, or they're just like stressed, or they're kind of using their muscles. You can see this example of this EEG, or like they're tense, for example. See this. So it's kind of like you kind of know how to read their brain in a way, but not really read their minds. It's just what they're doing physically, whether they're awake or asleep. EEG is good for that, and on the right hand side, you'll see a normal EEG where the patient is relaxed, and you will see, if you compare it to the left hand side, you'll see there isn't really much of a muscle activity. That's because the patient is not agitated or not tense or not doing any physical activity. It just appears that the patient is relaxed, awake, and it's just showing brain activity recorded from the midline, and then you'll see the channels written here: FZ to CZ, CZ to PZ, PZ to OZ. So what these stand for is basically frontal, central region, parietal region, and O stands for occipital region. So this is the midline, and then it goes from FP1, which is the frontopolar one, and then F3, which is the left frontal. So if you see a number that's odd, there are in the left side of the head. So this channel, all the electrodes are left side of the head, and this one right here is the right side of the head. So all the even numbers, FP2, F4, C4, P4, O2, all of these here are on the right side of the head. So yeah, it's quite interesting how much you can read patients' body language through EEG. So the next slide we'll just see example of abnormal EEG where a seizure is happening. So on the left hand side here is an example of seizure. You can see that it is almost like an artwork. The brain is creating an artwork in the form of an EEG channels. So this is a myoclonic seizure where the patient is having myoclonic jerks. So if I were to play this video, unfortunately I don't have the video, but you'd see that the patient is having myoclonic jerks, and it is correlating with the EEG. So this would definitely be considered a seizure, and it's quite interesting because you can see the spikes here, right here, and it's kind of like spikes and then wave, spikes, wave, which are characteristics of epileptiform, which is usually what you see with epilepsy patients. So yeah, so spikes and then kind of like the wave. So this is known as the spike and wave here. So yeah, so this is a primarily generalized seizure where the whole brain is affected, as you can tell, like the whole channel right here, the whole brain is affected. And then, unfortunately, I don't have an example of a focal seizure, but what would essentially happen, focal seizure, you might see it being just on the one part of the brain. Let's say it's happening on the temporal brain, so you'd see it on the temporal region only, spike and wave discharges. So yeah, you'd see it on the temporal region and it stays there. So you'd see those discharges and then it stays there. And then on the right, it's also generalized, primary generalized seizure. Otherwise, in the past, they used to call it grand mal seizure or petit mal seizure. But the new term is primary generalized seizure. They also call it, there's a lot of names. GTC, generalized tonic-clonic seizures. But you can see at the opening, the patient stops counting, opens her eyes, and at this point, the patient is responsive, and looks like the limb movements is happening here. And slowly, so this is more like probably like I would say like a 10-second or 9-second seizure, because at the end you can see the seizure kind of like ending. Yeah, so, but it's interesting because you can see the artwork of the brain when seizure is happening. Unfortunately for the patients, at this point they're not at their best. And so putting everything together, the role of glutamate and seizures. So seizure induces elevation in extracellular glutamate. It contributes towards exotoxic damage. Chronic seizures can change the neuronal and glial expression of glutamate receptors and uptake transporters. This contributes towards epileptogenesis. And then GABA are neurotransmitters that usually blocks the glutamate activity. So if we were to make a guess, GABA would stop the spread of seizures. But unfortunately, during a seizure, GABA is something that is inhibited or reduced. And as a result, more NMDA receptors are activated or overexcited. So you'll see influx of sodium and calcium in presynaptic terminal, and more glutamate is released in the synaptic space. And as stated before, GABA is inhibited, NMDA receptors activated, overexcited, hence more glutamate is released in synaptic space. And there's a flowchart on the left-hand side which shows the representation of role of glutamate and seizures. In summary, overexcitement of receptors of glutamate and inhibition of GABA, which leads to seizure. And just to recap, glutamate is the primary excitatory neurotransmitter responsible for spreading seizures in the brain. And just a couple minutes ago, I went over the two types of seizure: primary, generalized, and then the focal seizure. And just to keep in mind, focal seizure could become primary generalized. Seizure-induced elevations in glutamate receptors, leading to exotoxic damage. Seizure can be very difficult to control if not treated. And another thing I just want to mention, uh, anything, any seizures more than 5 minutes is very life-threatening. Um, at that point, I would say uh, should be brought to ED or just to the hospital as soon as possible. Um, yeah, so that's the summary. And for the discussion, I just want to, or future directions, I just want to talk about what we could do in the future, how to stop the spread of glutamate, which is responsible for initiation and spread of seizures in the brain and brain damage following a postictal state happening during a prolonged seizures or recurrent seizures. So how do we stop that? So we have some idea on how to control it. We use anticonvulsant medication such as carbamazepine, phenytoin, topiramate, lamotrigine, valproic acid, zonisamide. They work by calming the hyperactivity in the brain, which is what's happening during a seizure. The whole brain is just like super hyperactive, causing a seizure. So hence some of these drugs you'll see, these are used to treat ADHD, epilepsy, prevent migraines and other brain disorders. And these drugs work to stabilize the receptors by inactivating it. Other things I kind of want to investigate maybe in the future. Sometimes there is no known cause for a seizure. There is no trigger, there is no symptoms, but patient, they keep suffering from seizures. They never had anything, like no accidents, because a lot of the times people could get into accidents, hit their head, and have seizures. If they have subdural hematoma, that could cause seizures. Sometimes none of those. Some people could be completely healthy, and then all of a sudden they have a seizure. So what's exactly causing it? This is something I want to look in the future, whether through as a graduate student or a medical student or as a medical doctor. And then I want to find more side effects of seizure drugs. I know there's more side effects from seizure drugs. I can see it from my patients sometimes when I go to their room. There are a lot more symptoms of these drugs. So that's something I could look into it in the future. And something I just want to add, due to recent pandemic, I've seen some of the patients experiencing seizures from COVID-19, so this is something also I could, investigate in the future. What's causing the virus to induce a seizure or provoke a seizure. So that is something I want to look at in the future. Here's my bibliography. Thank you very much for listening. Sorry I went over 10 minutes. Thank you very much.